สวัสดีครับ Hello. Thank you for the opportunity for me to give an update on my research project under the support of the Mid-Career Grant from NRCT. My name is Thanet Kangsamaksin from the Department of Biochemistry, Faculty of Science, Mahidol University. And the title of my research project is the roles of lymphangiogenesis in cancer-associated lymphedema treatment. So let me give the introduction to our research problem first. So since we know that cancer patients who receive treatment such as lymph node dissection, surgery or radiation often suffer from the condition called lymphedema, which is the tissue swelling due to localized fluid retention. And this is generally caused by a compromised lymphatic system. And there are several physical or physiological treatments of lymphedema, and they prove to be somewhat successful but rather short-lived. So there's a problem to these cancer patients. The examples of the physical and physiological therapeutic approaches for lymphedema include the compression garments, complex decongestive physiotherapy, or even liposuction. So as you can see that this can be a very discomfort to the patients. And also physiological therapeutic approaches include the anastomosis of lymphatic vessels or lymphatic and venous vascular structures, and also vascularized lymph node transfer, which is a transfer of the lymph node from the omentum to the site of lymphedema or the site of swelling to release the fluid retention. And this is a research question that we received from the clinic, in that the surgeon who performed this technique would like to improve the outcome of the patients. And we set up the research questions in that we would like to understand the process of lymphangiogenesis, especially in the context of the lymphedema treatment. And that lead to our objectives of this project. First, to uncover the crosstalk and overlapping nature of angiogenesis and lymphangiogenesis. Second is to characterize the lymph node that were transferred to the patients and investigate the possible roles in the lymphatic endothelial cell biology. And last is to elucidate the role of inflammation in the process of lymphangiogenesis. And here is the experimental outline as flowchart. As you can see that there are experiments that try to address three parts of the project. So first, let me update about the results from the first part of the project, which address the crosstalk between angiogenesis and lymphangiogenesis. So we would like to investigate the effect of blood endothelial cells in lymphatic endothelial cells. So we observe the characteristics of lymphangiogenesis process by breaking it down to three parts, which include LEC activation, LEC progression, and LEC morphogenesis, and include several assays to try to address the specific steps in lymphangiogenesis. So first, we want to collect the blood endothelial cells by using HUVEC as our model. So we collect HUVEC derived factors by collecting the conditioned media and treat LECs with these conditioned media and try to see what would be the effects on this LEC's biology. So first we look at LEC activation, and we found that the conditioned media had no impact on cell adhesion, but it significantly decreased the rate of cell proliferation. on a similar trend, we found that the conditioned media significantly decreased the rate of cell migration from the wound healing assay as well. And with these two sets of experiments, we found that the beginning stage of lymphangiogenesis seemed to be affected by the presence of HUVEC-derived factors. So next we look at the LEC progression and also LEC morphogenesis by looking at the tube formation assays. So basically we seed LECs in matrigel, and these endothelial cells will start to change their morphology and adapt to the tube formation structures. And we found that HUVEC conditioned media seem to increase the ability of these LEC cells to form the tube-like structures, which is very interesting because we found in the previous slide that it kind of slowed down the proliferation and cell migration. But when we look at the tube formation, this condition seems to help or promote the process of tube formation in LECs. And when we look next to at the LEC identity by observing several LEC markers, including PROX1, LIFE1, and CDH5, which is VE cadherin, we found that this conditioned media significantly decreased the expression levels of these LEC markers, suggesting that the HUVEC conditioned media might destabilize or decrease the identity of LECs, and that fits with the earlier results that we found, and this may explain why the process of lymphangiogenesis is promoted, in that because LEC is destabilized, and when LEC is less mature, it has more opportunity or more chance to form tubes or to reconnect with each other and form a better network. And that's what we found with LEC tubulogenesis. So after all of these results that we found, we also want to see whether the HUVEC conditioned media would affect the function of the newly formed LECs or newly formed lymphatic vessels by looking at the LEC integrity and also permeability by observing the sonulin-1 protein, which is a tight junction protein, and this protein should be localized in the cell membrane where cells are connected. And we found that the HUVEC conditioned media did not significantly affect the expression levels as well as the localization of these tight junction proteins. So it means that the HUVEC conditioned media or HUVEC derived factors might not have any impact on the functions of LECs. So to summarize what we have done so far in this part, we finished all the LEC assays and we found that the HUVEC conditioned media or the presence of blood endothelial cells may promote the process of lymphangiogenesis by destabilizing the lymphatic endothelial cells and also making them less mature when compared with the control. And that is very interesting to us. So now we are in the process of investigating the experiments in AIM-2, which is to elucidate to characterize the lymph nodes function and their roles in lymphatic endothelial cells by collecting the lymph nodes from patients and also performing proteomic analysis and also gene expression analysis, and this is in progress. And this is also described in this flow chart, the next set of experiments that we try to address. So the output and the outcome for this project, we propose to have 3 publications and 1 master's graduate. We have one manuscript written and prepared to submit, and hopefully by the end of the project we will have 3 publications as proposed. And we have one undergraduate student applying to continue with us with this project. So hopefully by the end of the project we will have 1 master graduate as promised. So with the problems and obstacles, I think it's very similar to other projects within these past 2 years. We have the COVID-19 pandemic and the lockdown of the labs. However, we adjusted our project and tried to reschedule the experiments and finish most of the in vitro parts as best as we can. And hopefully we will still finish the project with the same output and outcome as proposed. So last, I would like to thank all the lab members and collaborators for this project, especially Mr. Krishnapong Daidaskon, our undergraduate student who did our senior project on this project, and Ms. Ratana Winanhan Chaina, our scientist, for great help with the setting up experiments and guiding Krishnapong throughout his senior project. And also we would like to thank our collaborators from Washira Hospital, Dr. Warapot and Dr. Chakrit, and Dr. Nontawat for helping with the patient sample and clinical data. And last, I would like to thank the financial support from Mahidol University and NRCT, the Mid-Career Grant. Without this support, this project would not be possible at all. Okay, thank you very much for listening, and I would like to take on any questions that you may have. Thank you.